Infection and defects in the mechanisms of defense and alterations in immunity. First, we're going to talk about the factors for infection. The term communicability refers to the ability of a particular pathogen to spread from one individual to another, which will cause disease. So think about, in terms of measles or pertussis, this has a high communicability because it spreads very easy. HIV is of a lower communicability. So some diseases are highly communicable, they spread easily from person to person, and others have a lower communicability and do not spread as easily. In terms of infectivity, this describes the ability of a pathogen to invade and then multiply within the host's body. And the ability to invade and multiply in that host is caused by pathogens first being attached to a specific receptor on the surface of that host cell, and this is going to initiate the infection. They may release enzymes that then further facilitate tissue invasion, and it allows the pathogen to establish itself within that host. Now, successful pathogens, they can escape from the phagocyte. Remember, they're the immune cells that are responsible for destroying invaders. And they can also spread through the lymphatic system and the bloodstream to reach different tissues throughout the body and different organs throughout the body. The term virulence refers to the capacity of a pathogen to cause severe disease in the host. For example, we'll go back to measles. This is of low variance. Rabies is highly virulent. So different pathogens have varying levels of virulence, and it ranges from mild to very highly virulent. In contrast, if you think about measles being low virulence, self-limiting, mild symptoms, rabies is highly virulent and it has severe neurological disease manifestations, and it's almost always fatal if it's not treated promptly. Therefore, virulence factors of pathogens can include the production of toxins, the ability to invade the immune system, or the ability to damage the host tissues. So being able to understand the communicability, infectivity, and virulence is going to be really important as we continue talking about factors of infection and how healthcare professionals need to develop a strategy for prevention, control, and treatment. So we're going to also talk about pathogenicity, portal of entry, and toxogenicity. In terms of pathogenicity, this is the ability of a microorganism or other invading agent to produce a disease in a host that is susceptible. And the success of that pathogen in causing disease really depends on various factors, and that can include communicability, infectivity, and the extent of damage, as well as the virulence. When we talk about the portal of entry, this is where the pathogen actually enters the host body and establishes the infection. And different pathogens have specific portals of entry that are very characteristic of that pathogen and their mode of transmission and characteristics. So let's talk about the common portals of entry. These include direct contact, which means that the pathogen is going to enter the host through direct physical contact with the infected individual or the contaminated surface or surfaces. Another common portal of entry is inhalation, and this is the airborne transmission. So airborne pathogens will allow themselves to enter the respiratory tract and cause infection. By ingestion, this could be through the consumption of food, water, or any other objects that are contaminated and that have the ability to introduce the pathogen in the GI tract. And then lastly, it could be an animal bite or an insect bite, and Think about mosquitoes or ticks. The term toxogenicity, refers to the ability of a pathogen to produce a toxin, a soluble toxin, also called an endotoxin, which greatly influences its degree of virulence. This is going to contribute to the development of that disease. The soluble toxins are released by the pathogen, and they travel through the body, and they're going to infect all the tissues and the organs as they're moving through. Endotoxins are components of the outer membrane of the certain bacteria, and it can elicit a harmful effect when released during bacterial cell lysis. So I want you to think about the process of infection. What really does it include? And we'll discuss this further in class. The process of infection includes the colonization, the invasion, the multiplication, and dissemination of a pathogenic microorganism. So what does this mean? There's potentially many pathogenic microorganisms that are in the natural microbiome that cause severe disease. And our human bodies have a symbiotic relationship with the normal flora maintained by the physical barriers. So think about our skin, our digestive tract, our respiratory tract, and this includes the complexity of microbiome, inflammatory, and immune systems. These microorganisms that can cause infection will do so if they can break through that protective barrier, or the defense systems are weakened in those areas. And so this is called an opportunistic microorganism. So basically, if we're going to have an infection, there needs to happen some physical damage to these tracts, so the GI tract, respiratory tract, etc. And this is just an example. And it can happen during trauma or surgery, and the bacteria may enter the bloodstream, potentially leading to sepsis. Sepsis leads to shock, and shock therefore leads to death. Think about the paper cut example. If we get a paper cut that contains Staph aureus, and this can cause a local infection of that area. The way our body responds to this may just be inflammation, although it could potentially also cause an abscess. Now, alterations in that microbiome by antibiotics may allow local overgrowth of opportunistic microorganisms. So think about Clostridium difficile, or C. diff. Immune deficiencies really allow an invasive systemic infection. So if our immune system and our ability to fight is low, then we are more susceptible to these opportunistic microorganisms. For more detail, I'm going to ask you to look at the table in the text, 10.1, Classes of Microorganisms Infectious to Humans. But we're going to take a look first at bacteria. Now, bacteria are prokaryotic, and this means that they're found in various environments in the human body, and they lack a nucleus or other membrane-bound organelles. So their characteristics are simple. They have a simple cellular structure, and they're small in size. So by definition, prokaryotes are a group of microorganisms that include bacteria, but unlike the eukaryotic organisms, prokaryotes don't have a true nucleus or membrane-bound organelles. So this is differentiating the difference between a eukaryote and a prokaryote. Prokaryotes are very diverse in terms of morphology, physiology, and their ecologic roles, and they can be found in various places: soil, water, extreme environments, think about hot springs, as well as within other organisms or pathogens. They can be classified based on their requirement for oxygen, meaning they are aerobic, or if they don't require oxygen, we can term this anaerobic. Aerobic and anaerobic, this helps us to describe the presence or absence of oxygen in the environment in which the microorganism, including bacteria as an example, can survive and can thrive. So let's listen to the difference between aerobic and anaerobic in a little bit more detail. Aerobic microorganisms, they need oxygen to carry out all their metabolic processes, especially for energy production. They have a metabolic pathway and enzymes that they can utilize oxygen as the final acceptor during respiration. They need oxygen for growth and survival. So some examples of aerobic bacteria include E. coli, Staph aureus, Pseudomonas. Those are aerobic. And the conditions that they are typically found in are well oxygenated environments, such as in the atmosphere, the upper layers of soil, and our human respiratory tract. In contrast, anaerobic microorganisms, they can survive and thrive in the absence of oxygen, or in environments with very low oxygen concentrations. They have an alternative metabolic pathway. We'll call this the anaerobic respiration or fermentation to actually produce their energy. These bacteria may use other molecules, such as nitrates, sulfates, as their acceptors instead of oxygen during respiration. Some anaerobic microorganisms are very strict anaerobes and can't tolerate any oxygen at all. And so while others can use small amounts, some cannot use any. And there's actually some that can switch from aerobic to anaerobic depending on the oxygen availability. Some examples of anaerobic bacteria are C. diff. That's probably the main one that we'll talk about today as being anaerobic. So again, anaerobic conditions are environments with limited or no oxygen. So think about the very deep layers of soil, think about our GI tract, and other areas in our body where there's very poor blood supply, or areas of our body that may have now have poor blood supply due to disease. Lastly, we're going to talk about bacteria in terms of whether they are gram-positive or gram-negative. And gram-positive bacteria, remember that staining process that you used in microbiology, where you put the dye, and if it was gram-positive, that crystal violet iodine would make the bacteria appear purple when looking under the microscope. Well, the cell walls of a gram-positive bacteria are thick peptidoglycan, and this is what retains the dye. So Staph aureus, Streptococcus, C. diff are gram-positive. Gram-negative, they don't retain this crystal iodine dye. And so when you're looking at the microscope, it does not have that purple layer. They do have a peptidoglycan layer, but it's very thin and it won't retain the color. And so it's not going to retain that dye. These gram-negative bacteria can be counterstained with red dye, and so they appear pink or red under the microscope. Examples of gram-neg bacteria are E. coli, Pseudomonas, and Neisseria gonorrhoeae. We're going to talk a little bit more about Staph aureus being our example of bacterial disease. This is a very specific type of bacteria, and again, it belongs to the gram-positive group. It's found in the skin, the nasal passages, and it can be part of somebody's normal flora, but it can also cause infection if given the right circumstances. In terms of the pathogenicity and characteristics, Staph aureus has the potential to cause severe life-threatening infections, and we'll call this hospital-acquired or nosocomial infections. They're often associated with surgical sites, IV catheters, or ventilator use. And it can be considered an opportunistic pathogen, meaning it can cause infection and the host's immune defenses are compromised, or when the host immune systems are compromised. And the ability of Staph aureus to form these biofilms really contributes to its ability to colonize on surfaces. So it makes it very resistant to antimicrobial treatments, and they're more difficult to eradicate. The bacterium will secrete an exotoxin, and this is a potent toxin that can damage the host tissues and contribute to the development of the disease. And on the next slide, we'll talk a little bit more about exo versus endotoxins. Antibiotic resistance is a significant problem with this bacteria, with Staph aureus, and it's developed resistance to multiple antibiotics, including methicillin-resistant Staphylococcus aureus, or MRSA, and our group presentations will talk about this in more detail. Okay, so exotoxins, these are enzymes that are produced by certain bacteria, and these potent toxins have the ability to damage the plasma membranes of the host cell. They disrupt the integrity of the membrane, leading to cell lysis and even dysfunction. And additionally, the exotoxins can interfere with essential enzymes involved in the protein synthesis that's needed within the host cells. And by disrupting these vital cellular processes, these exotoxins contribute to pathogenesis of the infection. However, on the other hand, endotoxins are components of the cell walls of certain gram-negative bacteria, and these toxins are released when the bacteria undergo lysis or cell death. Endotoxins have the ability to activate the inflammatory response in the host. Let's say this one more time. Endotoxins have the ability to activate the inflammatory response of the host. And so this will stimulate your immune cells to produce pro-inflammatory mediators. They're going to recruit additional immune cells to that site of infection, and they can also include the production of fever. And this is an important defense mechanism in your body's overall defense against infection. Bacterial pathogens will provide a variety of toxic molecules, and they may kill the individual cells, they can disrupt the tissue, and they can protect against inflammation. Differentiating between the two is important. Exotoxins are proteins released during microbial growth, and they have a very specific or they can have very broad effects on the connective tissue and the cell. They are also classified by their mode of action. So type 1 toxins bind to the cell and they alter cell function, and type 2 toxins, they cause the damage to the membrane of the cell. And type 3 toxins, they can actually enter the cell. So we're talking now again about exotoxins. A little bit more about endotoxins. Um, they can be released during bacterial growth and during the treatments with antibiotics. So, antibiotics cannot prevent the effects of endotoxin. Bacteriemia refers to the presence of bacteria in the bloodstream, or septicemia. And this indicates the growth and multiplication of bacteria within our bloodstream. And this happens when our body's own defense mechanism fails to contain or eliminate a bacterial infection. Let's give, for example, gram-negative bacteria. These are the ones that are often responsible for causing bacteremia or septicemia. The bacteria of gram-negative bacteria, they have cell walls that have endotoxins. And remember, they're going to be released into the bloodstream when bacteria undergo the lysis or the cell death. These endotoxins have profound effects on the body's immune and inflammatory systems. When these endotoxins are released in the blood, they can activate the complement system and then the clotting cascade. And this activation of the complement system will then lead to the production of various mediators that will furthermore enhance the immune response. But it can also contribute to the development of further tissue damage. That clotting cascade can result in the formation of blood clots, and this can also compromise overall blood flow. So let's think about the significant consequence of endotoxin release. It's going to increase the capillary permeability, and this increased permeability is going to allow a large volume of plasma to escape from the blood vessel and then move into the surrounding tissues. That plasma volume can lead to hypotension, and in severe cases, it can also lead to cardiovascular shock. And we know that this can have serious consequences for organ function and then overall our hemodynamic stability. The most common affliction of humans refers to an infectious disease that affects a large number of individuals. This is a viral disease. These diseases are caused by various microorganisms and can have significant impacts on global health, as we've seen with the COVID-19 virus. The replication of this infectious agent depends on its ability to infect and multiply within host cells. And it's crucial for this microorganism to establish an infection and cause disease. Different pathogens have very specific microorganisms to invade and exploit the host cell, and this allows them to replicate and spread all throughout the body. Now, despite being simple organisms, the pathogens can cause a wide range of symptoms and diseases in humans. They can affect various tissues and various organs, leading to localized and systemic infections. However, many viral infections are self-limiting. This means that the body's immune system is capable of controlling and eliminating this infection over a period of time. They can be transmitted through different routes. Aerosol transmission means that it's going to involve the dissemination of the infectious particles through the air. Typically, this is going to be a respiratory droplet, and it's going to be expelled by coughing, sneezing, or talking. Infected blood can also serve as a source of transmission. Think about blood transfusion, needle sharing, intravenous drug use, or even an accidental needle stick injury in the healthcare facility. Sexual contact can also be a way of transmission, and this is through the exchange of bodily fluids. And then vectors. Vectors are mosquitoes, ticks, fleas, anything that can carry and transmit the pathogen from one host to the other. The first step in viral life is attachment, and we're going to go through the stages of viral replication, and they're not capable of independent reproduction. They need a permissible host first. So with attachment, this is mediated by a viral surface protein that interacts with the complementary receptor of the host cell. Now, after it attaches, that virus is going to penetrate the host cell either through direct fusion of the viral envelope within that cell membrane through a process called endocytosis, and where this virus is engulfed by the host cell and enclosed in the vesicle. That's attachment and then penetration. Now, once it's inside the host cell, the virus undergoes uncoating. And this is the removal of the viral capsid, or that envelope that's going to release all the genetic information of that virus. This is going to allow the viral genome to be accessible so that your body can replicate it and transcribe it. And during this replication process, the viral genome will utilize the host cell, and it's going to produce viral proteins and replicate the genetic material. It's all using the machinery in that host cell. The viral genome then is going to integrate into the host cell, into the host cell's genome, and then utilize the host cell's own transcription and translation process to generate a new viral component. And after replication, the viral components are going to be assembled back into a new virus or new viral particles within that host cell. The assembly process will package the new genome, the synthesis of the viral proteins, and they're going to assemble it back into complete viral particles. They're putting it all back together. And then now we have this newly formed viral particle that's going to be released from the host cell through various mechanisms. Some of them may be lysed or destroyed by the host cell, and this is going to lead to more viral particles. Other viruses can bud off that host cell, and they can acquire their own envelope, which will have their own cell membrane. Now, after the replication and assembly of the new viral components, the copies of the viral genetic materials are going to be made within that host cell. This process involves the synthesis of new viral RNA or DNA molecules. Once the viral genetic material is replicated, new virions or complete viral particles are released from that infected host cell to infect other host cells. And this can occur through various mechanisms such as cell lysis. And we know cell lysis, when this occurs, that cell is ruptured. Or it can happen through budding, where newly formed viral particles acquire, again, an envelope on its host cell all by itself. During latency, the viral genome remains within that cell without actively replicating or producing new virions. Then these latent viruses can persist in the host cell for long periods of time. They don't cause any symptoms, they don't cause any issues. But then there's a certain trigger, such as stress, or there's hormone changes in the body, or maybe there's an underlying disease that can reactivate this latent virus. And this can lead to the production of new viral particles being developed, and then we'll have more symptoms and more disease. So think about a virus that can enter a latent state, such as herpes. We can have a recurrent cold sore or genital herpes outbreak, or we can have a very latent period where we don't see any signs or symptoms. Inhibition of the host cell, the DNA, the RNA, or protein synthesis, we actually will have viruses that can interfere with the normal processes of our own replication of DNA and our own transcription of RNA and our own synthesis of protein. And this can disrupt the host cell's machinery, and it can be utilized to help the virus replicate instead. So it's interfering and disrupting the normal cellular functions, and this is what's contributing to the viral symptoms that you might see. Certain viruses can disrupt the lysosomal membrane within the host cells, and some viruses have the ability to transform infected host cells into cancer cells. These viruses are known as oncogenic viruses, and they can alter the host cell's genetic material. They can cause this to lead to uncontrolled cell division and formation of tumors. So an example of an oncogenic virus would be HPV, or the human papilloma virus, Epstein-Barr virus, which is a very common virus, abbreviated as EBV. They're associated with cervical cancers and other types of lymphomas as well. So HPV for cervical cancers and EBV for lymphomas. Viral infections can weaken our immune system and they can damage the host tissues. So they create this environment that's very favorable for secondary infections or secondary bacterial infections. So think about maybe if you've had a recent illness that was first considered to be viral, and you went to go see your primary care provider and they didn't give you any medications and told you to go home, to rest, to drink more fluid. There's nothing we can do because it's a virus. You let it play out. However, this virus can facilitate colonization and growth of bacteria. So later on, it can lead to bacterial infection. So then the same illness, you might need to go in later and get antibiotics to help to treat the now more complicated and severe disease manifestations. Now, viruses have evolved and they have various strategies to manipulate and destroy or disrupt a host cell and its function, including the fusion of infected adjacent host cells and the alteration of antigenic properties. These mechanisms will really contribute to the pathogenicity of viral infections. And here are two effects. The first one we talk about is the fusion of the infected adjacent host cells. And with this, some viruses can actually induce the fusion of the infected cell with the neighboring uninfected cell. And this results in the formation of a very giant multinucleated cell, or a giant cell, which we've talked about in the past. This fusion allows the virus to spread now more efficiently within the host, and it will evade the human immune surveillance because it's attached to an unaffected cell. Think about measles being one of these fusion type viruses. RSV, which is respiratory syncytial virus, is another one, and herpes virus is another example. In terms of the alteration of the antigenic properties, viruses also are able to modify the antigenic properties of an infected host, and this makes them appear foreign to the immune system. This alteration can then include an insertion of viral proteins on the surface of the host cell and the induction of changes in the expression of how that host cell surface molecules behave. The result is that immune system will recognize the infected cell as a non-self, and it's going to mount on it with the immune response that's going to eliminate that cell or that molecule. The immune attack can contribute to tissue damage. It can also lead to inflammation and inflammatory type symptoms. So what is antigenic? When we talk about antigenic variation, this allows the pathogen to change the surface antigen that are the targets of our protective immune responses. And so then the individual will develop protective levels of antibodies. And this pathogen will respond now by changing the antigens, which will then lead it to become resistant. So that's antigenic variation. The flu has an antigenic variation characteristic. This is why each year we need to change the vaccine that we use for the flu. They are known, the influenza viruses, they're known for their ability to undergo antigenic variation. And the seasonal flu vaccine has to be updated and administered annually because of this. We also think of it in terms of antigenic drift versus antigenic shift. Antigenic drift, this is going to be the minor genetic changes that occur gradually over time. The mutations affect the genes that encode the surface proteins of the virus. These surface proteins are primary targets of the immune response and the basis for the classification of the different influenza virus strands. This antigenic drift will lead to the emergence of new strains of the virus, and it's going to cause these slightly different circulating strains. The immune system's recognition and response to the drifted strains may be reduced compared to the previous exposure, leading to now an increased susceptibility of infection. Antigenic shift, this is referring to the major genetic change that occurs when two or more different influenza strains will affect the same host cell and change and exchange the genetic material. So this reassortment of genetic material can then result in the emergence of a novel influenza virus strand. And this has a combination of surface proteins from different viral sources. This shift is responsible for the reoccurrence of pandemics that include influenza A virus subtypes that emerge, and the potential to cause widespread disease really has little to no pre-existing immunity in the population because it's almost its own new virus. So this shift is relatively rare, but it can have a significant public health implication. We need a monitor and surveillance, we need monitoring and surveillance for these viruses worldwide so that we can detect and characterize any type of antigenic changes, whether it is a drift or a shift. So to sum it up, the drifts are going to be very minor changes and the shifts are going to be major changes. Which virus undergoes yearly antigenic drift, allowing for the emergence of new strains? So if you were listening for the last 5 minutes, you would have picked A for influenza. And the mutations are going to occur on the key surfaces of the antigens, specifically the H antigen and the N antigen. And this is going to allow for the emergence of new strains of the influenza virus. Now we're moving on to fungal infections, and these are caused by large microorganisms known as fungi. Unlike bacteria, fungi are eukaryotes, which means they have a true nucleus and they have other membrane-bound organelles. Let's look at the other differences. Their cell wall. Fungi have thick, rigid cells and cell walls that provide structural support and protection. So unlike bacteria, their cell walls do not contain peptidoglycans, which are the target of antibiotics, such as penicillin and cephalosporins. We use these antibiotics because they're able to penetrate through the peptidoglycans. But on a fungi, the unique cell wall makes it resistant to penicillins and cephalosporins. Because fungi belong to the eukaryote domain, along with plants, animals, and other protists, Eukaryotes have a more complex cellular structure compared to bacteria, and they will house their genetic material and other specialized organisms in this true nucleus. Fungi also exist in different forms, depending on the life cycle and the environmental conditions in which they're found. So some fungi, they actually exist as single-cell organisms, and if they're single-cell, we call them yeasts, and they can reproduce by just simple division or by budding. Yeasts are typically found they are multicellular structures called molds, and they're round and oval shaped. And they have long filaments called hyphae. And the molds will produce spores for reproduction, and they can form visible colonies on surfaces. So yeast, round and oval shaped, spores are the production of molds that can form visible colonies or surfaces, so you can actually see the molds. Fungi have diverse methods of reproduction. First, yeast reproduce by simple division, and their cells are dividing into identical cells. They can move from small buds into parent cells, and eventually they'll separate and become its own new individual yeast cell. Molds, they're going to be produced by spores. They're small, lightweight structures, and they can be dispersed by air or other means. And spores can germinate, and they can grow, and they can become new fungal colonies if they're under the right conditions. We can find them in our different body parts: skin, nails, our respiratory system, internal organs, and they can be mild or very localized, or they can actually become very severe and invasive. So treatments of fungal infections, unlike antibiotics for bacterial infections, we treat fungal infections with antifungal medications, and they target the specific aspects of that fungal life cycle. Decreases are caused by fungi diseases caused by fungi are called mycosis. They're very superficial, they're deep, and they're opportunistic. Mycosis can be characterized into three main types based on the depth of the tissue. They can be superficial, deep, opportunistic. And the superficial mycoses are located primarily on the outermost layers of our skin, hair, and nails. So most of the fungal infections that we'll see are superficial. The deep mycosis invasions will include the internal organs and the tissues. It can also be in our lungs or the bloodstreams. And lastly, the opportunistic mycosis. This will occur in an individual that already has a weakened immune system, someone with HIV or AIDS, or someone undergoing some type of immunosuppressive therapy. Fungal infections can be on the skin, hair, and nails, and this is called a dermatophyte. A dermatophyte infection results in a characteristic clinical presentation and are commonly referred to as ringworm. So you've probably heard that, and basically what that is talking about is just describing the circular, scaly red lesion that resembles a worm shape. However, it's important that we know that it's not really from worms, and it doesn't always have that worm-like shape. Instead, we use the term tinea. Tinea infections, this is caused by dermatophytes that are on our body, and we have three types that we'll talk about right now. Tinea capitis, this is anything on your head, so think of cap on your scalp or your hair, and this can cause hair loss, scaling, and inflammation to the head. Tinea pedis, this is athlete's foot, so think of pedal, your foot, and it infects the feet. Really, the most irritating manifestation is itchiness, peeling, and redness between the toes and on the soles of the feet. Tinea curis is commonly known as jock itch, and this infection occurs in the groin area, and you can see a red, itchy rash. Again, we'll use antifungal medications. Sometimes, if topical treatments are not working, you can resort to oral treatments. However, they can damage your liver, so you have to decide if first the person is a candidate for this type of antifungal medication, and if they are, you'll have to continue to monitor their liver function. Now we're going to talk about the pathogenicity of a fungal infection, and they are able to adapt to the diverse environments within the host. So they can tolerate a wide range of temps, from being in areas of extreme hot to extreme cold, and still be able to thrive. And that's why you'll see them in the different areas of your skin, hair, and nails. Additionally, certain pathogens can survive and replicate in environments with low oxygen levels, so the deep tissue or the abscess. To establish infection, this pathogen has to suppress the immune defenses of the host. So phagocytes, including the macrophages and the neutrophils, they are going to be the key immune cells that play the vital role in eliminating this type of fungal infection. However, some of the pathogens that can evade or impair phagocytic function, so they're not able to be destroyed by macrophages or neutrophils. Sometimes they can even inhibit the activity of the T lymphocytes, and we know we need the T lymphocytes in order for the cellular adaptive immunity to progress. Despite the strategies employed by a fungal pathogen, the immune system also has mechanisms to control and eliminate these fungal invaders. Phagocytes can play a crucial role in engulfing and destroying the pathogen through phagocytosis, and the T lymphocytes, which we know are the type of white blood cells that can coordinate an adaptive immune response and directly kill through the C toxic lymphocytes. The affected cell will produce cytokines to activate the other immune cells like the B cells. So the example of the fungal infection that we'll talk about is Candida Albions. This is a yeast infection, and it's a very common cause of a fungal infection in a human. It's also an opportunistic pathogen. So which this means is that this infection is usually caused when the host's immune system is being compromised, or the normal balance of the microorganisms in the body are being disrupted. Candida is a part of the normal microbiome, so things that already live on our bodies. It's commonly found on our skin, our GI tract, the vaginal area. But under normal circumstances, this Candida albicans is kept in check by our own immune system and other microorganisms. But when there's an overgrowth or imbalance of Candida, it can lead to this localized infection. And they're typically in specific areas, like the cutaneous candidiasis, which is on our skin, or we have a type called oral thrush, which is in our mouth, or a vaginal yeast infection in the genital areas. Those are the three most common types of Candida albicans. In individuals with a weakened immune system, so think of someone that already has HIV or AIDS, they're undergoing chemotherapy, thinking of another example, or organ transplant recipient, Candida can cause a disseminated infection, which means that it can spread to other areas and lead to the deep tissues, the organs, and even the bloodstream. So it can become very life-threatening when it is opportunistic. It can also have very high mortality rates, especially in critically ill patients or those that have an underlying medical condition that's very severe. They can invade the tissue and evade our immune responses. Now we're going to talk about parasitic infections, and these are caused by a wide range of organisms, including unicellular protozoa and larger worms known as helminths. This type of organism can be further classified into flukes or nematodes and tapeworms. Let's first talk about their symbiotic relationship. They have a symbiotic relationship with their hosts, and they benefit at the expense of the host, actually. They rely on it for nutrients, for a suitable environment so that they can grow and reproduce. A parasitic infection can be mild and range to severe. It just depends on the type of parasite, the extent of the infestation of the parasite, and their immune response of the host. So, for example, protozoas, they are unicellular organisms, and think of parasites such as malaria or amoebas. Malaria is caused by Plasmodium parasites, and this is a significant global health concern that we're trying to eradicate. Usually malaria is found in tropical and subtropical regions. And then in terms of amoebic infections, such as amoebic dysentery, they can cause GI type symptoms and lead to complications if they're not being treated. Let's see, anything else here? To talk about the parasitic infections being spread from human to human, but they can also be spread through vectors. Again, those are mosquitoes, flies, or ticks. The transmission can occur through various routes: contaminated food or water, infected individuals through body fluids, or the bites of the vector organism. Parasitic infections also lead to tissue damage that can occur directly through the release of toxins by the parasite or indirectly through the inflammatory immune response of the host itself. So now we're going to talk about the countermeasures that we have, or in other terms, infection control measures. So parasitic infections, they pose significant challenges to our public health, as well as fungal, viral, and bacterial type infections. And so we have to really work on the environmental measures like waste disposal, water treatment, contamination prevention, food safety, vector control, using the correct insecticides, hand washing, really ensuring access to healthcare facilities and being able to offer appropriate treatments is going to make it or break it in terms of infection control. We also know that vaccines play a crucial role in preventing certain parasitic infections, bacterial infections, viral infections. So we'll continue to do more research and surveillance, and we also have international collaborations to help organize and to help research and partner together to find better solutions and to improve health outcomes. Antimicrobials, these are substances that will inhibit or kill a microorganism. So the one that we know most is like an antibacteria. Or an antibiotic. When we talk about bactericidal antimicrobials, these are ones that are going to kill the bacteria by disrupting the cellular process itself. And this is going to target the cell wall or the cytoplasmic membrane or the nucleic acid synthesis or protein synthesis. And even it can be through the energy metabolism. So each bacteria has its own weak defense, and the antibiotics will have its own strength. And so we try to match the antibiotic that has the strength against the weakness of the bacteria. In terms of bacteriostatic antimicrobials, these are the ones that are going to inhibit the growth and production of bacteria. So it doesn't really kill them directly, but it's going to limit their ability to reproduce. And it's going to interfere with this process, which will then slow down the infection. And then in terms of inhibition of cell wall synthesis, antimicrobials will interfere with the bacterial cell wall itself, and it's very essential for maintaining the cell structure and integrity. So if we can inhibit the cell wall synthesis and weaken the bacterial cell wall, then we can promote cell lysis and cell death. We can also try to damage the cytoplasmic membrane, and this is used to disrupt the structure and the function of that membrane, and then this will compromise the integrity of that membrane, and so it will leak all the essential cellular components, and then eventually that cell will die. And then we also have antimicrobials that can interfere with the replication. So it's really being targeted to the nucleic acid metabolism, so the DNA or the RNA. And so we target these processes, and this is going to disrupt that genetic machinery, and so the bacteria can't proliferate. Or we can inhibit the protein synthesis. This is going to target the bacterial ribosomes, which are responsible for protein synthesis. It's going to bind to the ribosomes and interfere with the translation process. And then we also have interference with folic acid metabolism. Folic acid is essential for bacterial metabolism and the synthesis of DNA, RNA, and proteins. And so if we can inhibit this enzyme that involves folic acid metabolism, then we can disrupt and hinder the growth. We can also modify the metabolism. If we can disrupt the bacterial energy production by targeting the enzymes involved with the energy metabolism pathway, such as glycolysis or electron transport chains, then we can interfere and cause an impairment in the bacterial survival and replication. Bacteria can acquire resistance to antibiotics because it also, the bacteria can go through genetic mutations, and they can occur spontaneously in the bacterial DNA. These changes in the target site of the antibiotic or the production of the enzymes will then inactivate the antibiotic itself. And so some bacteria possess enzymes such as beta-lactamase, and this can chemically modify the antibiotic and then make it ineffective. So the enzyme will break down the antibiotic, and it won't be able to reach that target area. And if it can't reach that target area, it can't exert that antimicrobial effect. We're going to talk about penicillin resistance because this is a major one, and it's our prime example. Through the production of beta-lactamase enzymes, they can degrade the penicillin. And so the bacteria will develop alterations in the structure of their wall. And this is where the penicillin binding proteins are. And it makes them less susceptible. And the action of the penicillin is not going to be very strong. Bacteria can modify the target molecules that antibiotics bind to, and then this makes them less susceptible to the effects of the drugs. For example, if you have a mutation in bacterial ribosomal proteins that reduce the binding affinity of antibiotics, this can target the protein synthesis. And then some bacteria have efflux pumps, and this can actively pump antibiotics out of the bacterial cell. And so if it's pumping it out, it's reducing that intracellular concentration of the drug, and it's also going to be limiting its effectiveness. But what and why do we have antibiotic resistance? Well, we have a lack of compliance with the therapeutic regimen, meaning that sometimes patients are not taking the correct dose, they're not using the correct antibiotics for the right bacteria, or they take an incomplete dose or regimen of the fully prescribed course. So they might skip a dose or take it too short or take it too long. This allows the bacteria with intrinsic or acquired resistance to then survive and multiply. Antibiotic resistance also allows bacteria that are more naturally resistant to have acquired resistance through flourishing. And this is not affected by the antibiotic being used. The selective pressure promotes the growth and spread of resistant bacterial strains. This is called selective resurgence. We also have the overuse of antibiotics. So overuse and misuse, this is using a bacteria for viral resistance. So again, going to the PCP with the viral infection but wanting an antibiotic, patients are persistent, your PCP prescribes it, and so now we've overused and we've misused this antibiotic. Well, this excessive use of antibiotic increases the likelihood of encountering resistance, and it provides an environment where the resistant strains can now thrive. And then also antibiotics have a target pathogenic bacteria, but they also disrupt the normal microbiota in the body. So this disruption can lead to now the overgrowth of antibiotic resistant bacteria and again promotes the development of resistance. Vaccines are biological preparations that contain a weakened, also termed attenuated, or inactivated or dead pathogen of the recombinant viral protein. They're designed to stimulate the immune system to produce an immune response against specific pathogens. So we have long-lasting immunity, and this is when vaccines are formulated to stimulate the immune system to recognize and remember specific pathogens. The immune response leads to the production of antibodies and the development of memory cells. This provides long-lasting protection. We use CDC schedules or recommended vaccination schedules depending on different age groups. So infants have their own schedule, children, adolescents, and even adults have their own schedule. And this is based on the evidence that has shown that these ages and stages of life are the recommended timings and that they have worked optimally to protect us against vaccine preventable diseases. Vaccine development is expensive, it's complex, it's costly. We have to go through the extensive research, preclinical testing, clinical trials, and regulatory approval. Although we've seen that some of these things can be expedited during a pandemic. The cost associated with the vaccine is not only including research and development, but also the manufacturing and then the safety and efficacy of the vaccine. But as you've seen through this pandemic, there has been a reluctance to vaccinate. Despite the benefits of vaccines, we still encounter vaccine hesitancy and a reluctance to vaccinate, and this can be due to various factors: misinformation, safety, philosophical beliefs, religious ideals, lack of access to vaccines. But we do have public health efforts that are focusing on providing the correct information and allowing our patients or our community to understand the importance of vaccination and prevent diseases. There are rare complications. Though vaccines are generally safe and very well tolerated, there can be severe complications. And so think about anaphylaxis and some of the questions that they may have asked you if you got vaccinated with the COVID vaccine. we're trying to understand, what the long-term effects of these vaccines are. And some of the newer ones, we don't have as much information, and therefore further research has to occur. Adverse reactions, if they occur, typically they're mild and temporary. Most often it's the soreness of the injection site or low-grade fever. Although we have seen incidences of more severe reactions in cases that are due to vaccination. So you can read those in the literature and also in recent news studies. Vaccines are biological preparations. They're designed to induce this long-lasting protective immune response. Some of them will last longer than others. Some will require boosters. Toxoids, these are specific types of vaccines that target bacterial toxins. And some bacterials produce these toxins, which are the primary cause of the symptoms and complications that are associated with that specific disease. So toxoids are prepared by inactivating or detoxifying the toxins, so they're no longer harmful, but they can retain their ability to stimulate an immune response. So when a person receives a toxoid vaccine, their immune system recognizes the inactivated toxin as the foreign substance, and then the body will produce an antibody to it. And so the antibodies can then neutralize the toxin if the person is ever exposed again to this type of disease. For example, the tetanus or diphtheria vaccines, these are actually toxoid vaccines. And then tetanus, if you don't know what that is, it's caused by the toxin produced by the bacterium Clostridium tetani. And diphtheria, this is caused by a toxin produced by, I'm going to butcher this, Corynebacterium diphtheriae. So you don't need to know that specifically, but do know that tetanus and diphtheria are examples of a toxoid vaccine. And then our common problems, we do have less access. We talked about compliance, unresponsiveness to the vaccine. Some, even though most of the ones that are put out to market are generally effective in inducing an immune response and they provide some type of protection, underlying health conditions, immunodeficiency, or genetic variations, they can contribute to the reduction in the response of the vaccination from individual to individual. In terms of vaccine resistance, this is the ability for the pathogen to evade even though the person gets the vaccine. And this can occur if there's genetic changes or mutations in the pathogen's antigens, and then the vaccine will be less effective. So we talked about the flu vaccine. And then reluctance, this is a term that we use if we have refusal to get a vaccine and it is driven by concerns about vaccine safety, efficacy, or necessity. And then we have passive immunotherapy. This is an approach that involves administering performed antibodies to provide immediate protection against the specific pathogen or the specific toxin. So we can use human immunoglobulins that contain antibodies against hepatitis. We used it for COVID, and they're used for post-exposure prophylaxis. And they provide passive immunity in individuals at risk for developing, we're using the example here, hepatitis, so it's at risk for developing hepatitis. But we also, again, we used it for COVID as well. We also used immunoglobulin and monoclonal antibodies. We use this for COVID, but in this example for rabies, the RIG is administered with the rabies vaccine, and this promotes immediate protection against the virus and anyone that's been exposed to this specific virus. Monoclonal antibodies are specific to rabies and can be used as passive immunity, providing targeted and specific neutralization of the virus. So it is an example of passive immunity. We also have monoclonal antibodies for RSV. And some people think that this is a vaccine that we give for RSV, but actually it's a monoclonal antibody. We give this to young children, mostly preemies, that are high risk for severe RSV infection, which is a respiratory infection. It's called respiratory syncytial virus. And these monoclonal antibodies are administered monthly during RSV season, and it provides temporary immunity and the reduction of risk for this infant or child to have severe respiratory complications. So, which vaccine contains a live virus that are weakened or attenuated? The MMR, or measles, mumps, and rubella varicella vaccine, varicella chickenpox, these contain live viruses that are weakened or attenuated, and they're able to continue to express the appropriate antigens, but only a limited and very easily controlled infection. So one current exception is the hepatitis B vaccine, which uses a recombinant viral protein. Vaccination against systemic toxins such as diphtheria and tetanus have been achieved using toxoids, and pertussis, or whooping cough, vaccines have been changed from a killed whole cell vaccine to a cellular extract or acellular vaccine, and that contains pertussis toxoid and additional bacterial antigens. So the answer here, the live attenuated or weakened, is B, measles, mumps, and rubella. Now we're going to talk about immunodeficiency, and this refers to a failure of the immune system's mechanism of self-defense, leading to increased susceptibility to infections and other diseases. So there's two types. There's the primary and the secondary. Primary meaning congenital, or secondary meaning acquired. So with primary immunodeficiency, this is a result of genetic anomalies or inherited defects of the immune system. And so they can affect components such as the B cells, the T cells, your phagocytes, your complement proteins. Primary immunodeficiencies are typically present from birth. And then sometimes, though, they're not manifested until later, but they're often diagnosed in childhood. They're caused by a single gene mutation, and they're relatively rare, but they can have significant impacts on the individual's health and the individual's quality of life. Secondary immunodeficiency, on the other hand, is acquired as a result of an underlying disease or an underlying illness. So think about the various factors contributing to immunodeficiencies like HIV or AIDS, or diseases like leukemia and lymphoma, or autoimmune diseases like lupus, even malnutrition or chronic diseases. Think about diabetes or kidney disease, and even medications such as immunosuppressants or chemotherapy. Secondary immunodeficiencies are more common than primary immunodeficiencies, and they can develop at any age. So clinical presentations of these immunodeficiencies will vary on the specific type of the underlying defect. So the common clinical features associated with the different types of immunodeficiencies are T cell or B cell or phagocyte deficiencies and complement deficiencies. The T cells may experience a higher susceptibility to viral, fungal, yeast, and atypical microorganism infections. These infections can be recurrent, they can be very severe, and difficult to treat. You might have persistent chronic infections, failure to thrive and grow, or delayed development if you have a T cell deficiency. With B cell and phagocyte deficiencies, this can impair the production of antibodies. And so phagocyte deficiencies will affect the function of the cell that engulfs and destroys the pathogen. And as a result, individual with this deficiency, they're going to be more prone to bacterial infections, specifically caused by microorganisms that require opsonization. And remember, this was the coating by the antibodies or the complement proteins. And this is making it attractive for phagocytes to come and break up that organism and engulf it. Common infections may include respiratory tract infections, ear infections, sinus, and skin infections. In terms of the complement deficiencies, this is a system that's part of the immune system that helps clear the pathogen. And so if we have a deficiency in the complement proteins, this can lead to an increased susceptibility to infections, particularly those that are encapsulated bacteria. So meningococcal, pneumococcal infections, those are examples of infections that are more opportunistic if you have a complement deficiency. Immunodeficiencies can be caused by various factors, and most of them are a result of a single gene defect. So, interestingly enough, the majority of immunodeficiencies are not inherited, but account for 25% of all cases. They manifest early in life, presenting in infancy, childhood, or they appear later in life. So we talked about those. They're rare, but they're decreasing, and you see the major groups or classifications here. With combined deficiencies, this is a result from underdevelopment of both T and B cells. And an example of this is SCID, which is severe combined immunodeficiency. This is a group of rare genetic disorders that will have severe impairment in the immune system's ability to function properly. And so there's either deficiency or there's an absence of T and B cells that are detectable within that adaptive immune system. This usually is accompanied by the underdevelopment of the thymus gland, and it can lead to absence or severely reduced number of functional T cells, because remember, the T cells mature in the thymus gland itself. And so with SCID, you have impaired cellular immune responses. You're not able to recognize or eliminate the intracellular pathogens. This makes individuals with SCID really highly susceptible for bacterial infections, viral infections, fungal, and opportunistic infections. In addition to T cell deficiency, we also see that there's a reduced level of immunoglobulins or antibodies. IgM and IgA produced by B cells can be absent or significantly reduced with SCID. We also have bare lymphocyte deficiency. This is known as a MHC class 1 and 2 deficiency. And remember, we need these complexes to attach, or these molecules to attach. They play a crucial role because they present the antigen to the T cell, and this is going to help this immune function to occur properly. In the bare lymphocyte deficiency, they are without, or they're bare of the MHC molecule. So they may have enough B and T cells, but without the major histocompatibility complexes, or the MHCs, they're not able to bind together appropriately and be recognized as a pathogen. And so we have two syndromes, the Wiskott-Aldrich syndrome, which is an immunodeficiency disorder characterized by a triad of eczema, thrombocytopenia, and immunodeficiency. In this syndrome, there's a defect in the production of IgM, and this can lead to a depressed immune response, but also they have platelet abnormalities that can lead to bleeding. With DiGeorge, This is due to a 22q11.2 deletion, and it's a genetic disorder that is characterized by the underdevelopment or even the absence of the thymus gland. So they won't be able to develop T cells or mature T cells appropriately. Defective B cell development refers to a group of immunodeficiencies that affect the maturation and the function of B cells, and they're responsible for producing antibodies. These deficiencies can result in partial or complete absence of specific classes of antibodies. In the range of antibody deficiencies, we have defective B cell development that can affect either a single class of antibodies or it could be a multiple classes of antibodies. So IgG, IgM, IgE, IgD could be a combination of these antibody classes. They can also involve hypogammaglobulinemia, and this is a condition characterized by lower than normal levels of antibodies in the blood. So hypo, low, and then emia, meaning blood. Or it can be called agammaglobulinemia, and this is a more severe form of B cell deficiency when there's a complete absence of mature B cells and immunoglobulins. The deficiency is often caused by genetic mutations that disrupt the development of B cells in the bone marrow. We also have phagocytic defects. With chronic granulomatous disease, or CGD, this is a rare genetic disorder that affects the immune system's ability to fight infection because we don't have working phagocytes. We're missing a certain hydrogen peroxide system, which is crucial for the killing of pathogens by the phagocytes. So if we have defective phagocytic killing, this is because there's no hydrogen peroxide or other oxygen products that this phagocyte needs, such as neutrophils and macrophages. We have recurrent infections. So with compromised phagocytic killing, individuals with CGD are going to be prone to recurrent infections. And the most common types are going to be respiratory infections. And most of the time, they're going to have recurrent pneumonia. They can also have soft tissue infections, bone infections, abscess, and GI type infections. We also have granuloma formations, and these are tumor-like masses called granulomata. And these can develop in different organs, including your lungs, your skin, your bones, your liver, even your GI tract. And they're composed of a collection of immune cells, such as macrophages and lymphocytes, and are very characteristic of a person that has CGD. CMC, or chronic mucocutaneous candidiasis, this is severe recurrent candida infection, and it's due to a defective immune response to candida itself. The immune system is unable to mount an effective defense against the fungus, and it can lead to persistent infections, primarily on the mucous membranes and on the skin. So if you don't have enough C3b and C3a production, you're not able to activate C5 in the complement system. And so this can lead to life-threatening infections with encapsulated bacteria like influenza or streptococcus pneumonia. And this usually happens at an early age. Remember that the terminal component of C5 is necessary for the complement cascade. And so without it, in C5/9 deficiency, we have increased infections with Neisseria gonorrhea and Neisseria meningitis. So we need to have C3. We also have a deficiency in MBL, which is mannose binding lectin. And if you have that deficiency, you're missing the protein that's involved in the lectin pathway of the complement activation. Again, you put yourself more at risk for infection because it's also one of the proteins that are needed in the complement system. Microorganisms such as Streptococcus pneumonia, E. coli, Candida, they can all be more prominent in a person that has an MBL deficiency. When we talk about secondary deficiencies, we're talking about, or secondary immunodeficiencies, we're talking about acquired deficiencies or immune deficiencies that have developed over time because of another underlying condition or another factor. So the common occurrences are due to secondary immunodeficiencies having a more common than primary deficiency and genetic in nature. They develop in individuals of any age, and they can be caused by a variety of different factors. The secondary deficiencies are associated with underlying medical conditions such as HIV, AIDS, cancers, autoimmune disorders, malnutrition, stress, and also immunosuppressive medications can cause secondary deficiencies. Excuse me. In many cases, secondary deficiencies may not have a significant clinical impact or increase susceptibility to infections, but this is because the immune system is still functional to a certain extent. And over time, the individual may not experience frequent or severe infections. However, there's also variable severity. So they can be widely depending on what that underlying cause was in the first place and the extent of the immune system's impairment. Secondary deficiencies do not always increase susceptibility to infection. Individuals with these deficiencies may be more susceptible to certain types of infections, but associated with the underlying condition. So, for example, someone with HIV or AIDS may be at higher risk for opportunistic infections, but it doesn't mean that if you had a secondary deficiency that you're necessarily going to increase susceptibility to infections. There are specific ones. And really quickly, you're going to elaborate more on this in your med-surg class, but we'll talk about how we evaluate for immunity. We can get CBCs with differentials because we can look at our leukocyte count and determine if we have enough white blood cells, including the lymphocytes. We can also use flow cytometry. This is a technique used to analyze and quantify the different subpopulations of the lymphocytes. So we can look at T cells, B cells, and the natural killer cells. So we're looking at the composition and relative proportions of lymphocytes. This is going to help us to identify the type of abnormality to a specific cell population. We can also determine by quantifying the immunoglobulins, so also known as antibodies. And what we can do is use a blood test to quantify IgG, IgM, IgA, IgE, and IgD, and we can measure these levels in our blood. And we can also do an assay for the total complement system and check to see if we have all the components of proteins that we need to work together to mount an immune response in that complement system. These are just some examples of treatments We can use IVIG, which is an antibody that's rich fractions prepared for plasma are going to be pulled from many donors, and it replaces the person's antibodies temporarily. The half-life is usually 3 to 4 weeks, and we can also treat them repetitively. Bone marrow, umbilical cord transplants, umbilical cord cell transplants, fetal thymus tissue for thymic defects. Again, that could be found on DeGeorge syndrome as one of the types of disease processes that we might do that with. We can also do a mesenchymal stem cell injection or gene therapy. We can transfuse erythrocytes. So there are different types of treatments for immunodeficiencies. Now we're going to talk about acquired immunodeficiency syndrome, or AIDS, and this is caused by the human immunodeficiency virus, or HIV, which depletes your body's T helper cells, and it will specifically target the CD4 T helper cells. And this, if you remember, helps with both the humoral response and the cellular response of the adaptive immune system. And it can lead to destruction, resulting in a progressive decline in the number of T cells that are actually able to help with your immune system. And therefore, it's going to weaken your immune system. It can cause you to be more susceptible to opportunistic infections and certain types of cancers. In terms of retrovirus, HIV belongs to the retrovirus family, and this is characterized by the ability to convert its genetic information from an RNA into a double-stranded DNA, and this carries the enzyme called the reverse transcriptase, and it performs the conversion. So it allows that viral DNA to integrate into the host cell's genome. In terms of its structure, you can see the GP120 protein and the CD4 binding. In terms of that, the outer surface of the HIV or the human immunodeficiency virus is covered with glycoproteins, including the GP120. This protein will bind to the CD4 molecule, and it's primarily found on the surface of the T helper cell. This interaction between this GP120 and CD4, this is the initial step in where the viral entry process actually allows HIV to attach and enter the host cell. So if we have a destruction of the CD4 T helper cells, HIV can enter that CD4 T helper cell and replicate and spread throughout the body. And this replication cycle of HIV ultimately leads to the destruction of the CD4 cell, or the CD4 T helper cell. So the number of CD4 T helper cells is an important indicator of the immune status and the progression of HIV infection. So in a healthy individual, your typical CD4 T helper cell count is going to range from 800 to 1,000 cells per cubic millimeter of blood. However, with HIV infection, this can lead to a progressive decline of CD4 T cell count, and we will monitor this blood count at different stages to see if there's a need for antiretroviral treatment or if this antiretroviral treatment is working. CD4-CD8 ratio, this refers to the relative proportion of CD4 T helper cells to the CD8 cytotoxic T cells in the immune system. And in HIV infection, the destruction of the CD4 T helper cell leads to a decrease in this ratio. And the alteration in the ratio is a marker of immune dysregulation, and it tells us that there's progressive HIV disease. So some manifestations that you might see. You might see in the early stages of HIV that individuals may be serologically positive for HIV, but we don't see any symptoms at all. And this term is this is being termed serologically positive but asymptomatic. But once the T helper cells are below 200 cells, this is the diagnostic criteria for AIDS or acquired immunodeficiency syndrome. And this is made when the individual, which has an HIV infection, has a significant decline in the immune system, and that CD4 T helper cell, or sometimes just called T helper cells, is less than 200 cells per cubic millimeter of blood. This is a severe depletion of CD4 cells. In addition to CD4 T cell, the diagnostic criteria of AIDS is made in association with other clinical conditions and other lab tests. We're looking for atypical or opportunistic infections, such as pneumonia. These are also called AIDS-defining conditions. And additionally, we're looking at the antibody and detection through serological testing. So we're going to do an enzyme immunoassay, or an EIA. We can also do a rapid antibody test to confirm HIV infection. And this is typically, the confirmation is going to be through a Western blot analysis. So again, confirmation is through a Western blot analysis, and this is going to detect the specific antibodies for HIV. Some of the treatments, though you're going to go through them more thoroughly in MedSurg, are antiretroviral therapies, or ARTs, and they're a combination of therapies that are able to reverse the transcriptase inhibitors, the protease and the integrase inhibitors, including the fusion and the CCR5 agonists. And so these all have a specific part in HIV, and some of them, like the protease inhibitors, they're going to block the activity of the protease enzyme, which then prevents the production of mature and infectious viral particles. With the integrase inhibitors, they're going to target the integrase enzyme, and this is responsible for the functioning viral DNA. So you can see that all play a different part in the ART therapy or ART regimen. They have been known to reduce mortality, although there is no cure for AIDS. With effective treatment, people are living longer and they can have productive lives. In terms of variants and resistance, it's it's able to highly mutate. The rate is high, and it leads to the emergence of different strains and genetic variations of the virus. And so we have to regularly monitor the viral load and the drug resistance to identify any patterns and adjust the treatment regimen if needed. Our prevention strategies really are to control the spread of HIV, and this includes talking about safe sex practices, needle exchange programs, and using pre-exposure prophylaxis, also called PrEP. And this is when you take an antiretroviral medication before any potential exposure to HIV. And then it's recommended for individuals that are at very high risk for acquiring HIV. There's also PEP, which is the post-exposure prophylaxis, in contrast to PrEP being the pre-exposure. PEP involves taking antiretroviral medications after you've had a potential exposure to HIV, and this is supposed to reduce the risk of infection. It is recommended for any emergency situations, such as a needle stick or unprotected sex with a partner that you believe to be HIV positive. There are, sadly enough, cases of pediatric AIDS, and this is transmitted through pregnancy. It can be transmitted at delivery or through breastfeeding. It is very aggressive in children, and if you do not treat, they are most likely not going to survive past their second birthday. When you have a pediatric AIDS patient, usually you will see neurological symptoms like HIV encephalopathy, and it's very difficult to differentiate from the other risk factors. And you can also use antiretroviral therapy to treat pediatric AIDS as well. Now we're going to shift to hypersensitivity responses, and these are altered immunological responses to an antigen that results in disease or damage to the host. In terms of allergy, this is the deleterious effect of hypersensitivity to an exogenous antigen, so something out in the environment. In terms of autoimmunity, this is when your immune system mistakenly attacks and damages healthy body tissues and healthy cells. And normally this immune system is tolerant or self-antigens, and it recognizes them as part of the body, and it doesn't mount an immune response against them. But in an autoimmune disease, the tolerance breaks down and the immune system begins to target self-antigens, thinking that they're foreign. And this can it can result in chronic inflammation. You can see damage to organs and damage to tissue. So, for example, autoimmune disorders can include rheumatoid arthritis, systemic lupus, and multiple sclerosis. Alloimmunity refers to immune reactions against tissue or cells of another individual, but of the same species. And this usually happens in the context of transplantation. And so if you are the recipient, your immune system recognizes the transplanted tissue as being foreign, and it's going to mount an immune response and reject that transplant. We started last week talking about some hypersensitivity reactions, but we'll talk a little bit more in depth today. They are characterized by immune mechanisms type 1, 2, 3, and 4. A type 1 hypersensitivity, this is a reaction that's mediated by IgE antibodies, and it occurs when an individual is sensitized to a specific allergen and then re-exposed to that allergen again. But then when they're exposed, it activates mast cells and basophils, and you'll see various inflammation mediators. So you're going to see histamine come out and play. You're going to have an immediate hypersensitivity, which will include itching, swelling, redness, and in severe cases, anaphylaxis, which we heard about last week. Some examples of type 1 hypersensitivity reactions, IgE mediated, are allergic rhinitis, asthma, and food allergies. Type 2 hypersensitivity reactions, these will include tissue-specific reactions mediated by antibodies, usually IgG or IgM. The antibodies are going to target specific antigens that are present on the surface or cells of tissue and lead to the destruction or dysfunction of that cell. And it can occur through the complement activation, antibody-dependent cell-mediated cytotoxicity, or direct cell destruction. Common examples of type 2 hypersensitivity reactions include autoimmune hemolytic anemia, an Rh incompatibility between the mother and the fetus, and some drug-induced immune reactions. Type 3 hypersensitivity reactions are immune complex mediated, and they occur when there's an excess of immune complexes. Antigen antibody complexes, they're not being effectively cleared from the bloodstream and deposits in the tissue. And this will cause inflammation and tissue damage. So think about lupus being one of those, rheumatoid arthritis, and certain forms of vasculitis. These are all type 3 hypersensitivity reactions. And then the last one, type 4, this is cell-mediated. It involves T cells. And this occurs when sensitized T cells encounter an antigen and it leads to an inflammatory response instead. And the type of hypersensitivity is typically going to be delayed, and you might not see it peak for hours or even days after you were exposed. So, for example, a contact dermatitis reaction, like maybe a poison ivy, or maybe you touched something several days ago and then it didn't start manifesting into a rash until afterwards. Tuberculous skin test reactions, this is also a type 4 hypersensitivity reaction. or a graft reaction in an organ transplant. These are all types of type 4 cell-mediated reactions. So hypersensitivity, if it's immediate, minutes to hours, it's caused by the antibody, or it can be delayed several hours or maximum several days after re-exposure, and this is usually caused by T cell or a natural killer macrophage cell. In terms of anaphylaxis, though we talked about it, I'll just remind you it's the most rapid and severe immediate hypersensitivity reaction. It's systemic, it's generalized, it's cutaneous. Cutaneous meaning it's localized, and it can manifest in pruritus, itchiness, erythema, redness, vomiting, abdominal cramps, diarrhea, and breathing difficulties. So protecting the airway is going to be one of the major interventions for anaphylaxis. So although I did an overview of each type of hypersensitivity reaction, I'm going to get a little bit deeper here. Again, type 1 is the most common. It's IgE mediated, and there's a strong association to autoimmune and alloimmune diseases. Antihistamines will block the histamine receptor and control some of these type 1 hypersensitivity reactions, and so we try to use antihistamines if we see this type of IgE mediated reaction. The immune system is going to produce a very specific IgE antibody against the environmental antigen. This is known as the allergen. The allergen can then when it's exposed to the person with allergies, the allergen is going to bind the IgE antibodies. They're already attached to this Fc receptor on the surface of the mast cell. And remember, the mast cell is an immune cell, and this binding will then trigger that mast cell to release the inflammatory mediators, such as histamine, from their granules. So what is an allergen? Pollen, dust, pet dander, food, insect venom, those are types of allergens. So once that histamine is being released and it binds to now the H1 and H2 receptors that are on the various cells throughout the body. You even see H1 and H2 receptors on blood vessels, smooth muscles, mucus-producing cells. That activation is going to lead to an allergic reaction and all the characteristic symptoms: itching, swelling, redness, increased mucus production, smooth muscle contraction. Antihistamines are medications commonly used to manage the symptoms of allergic reactions. And so they are going to block that H1 receptor and reduce the effects of histamine. And that's what's going to alleviate the itching, sneezing, runny nose, etc. So the antihistamine is working on the H1 receptor. They can be over the counter. We can also prescribe them. They can be oral, they can be nasal, and they can also be eye drops. They're working on that H1 receptor. The most common in GI, foods, milk, nuts, wheat, eggs, shellfish. These are the most common allergens that can affect a GI type of response. In an individual with genetic predisposition to allergies, we call this atopy. Certain tests can be performed, and we can help to identify these specific allergens. And maybe some of you have had these tests done on yourselves. I've had a skin test done, and they put small amounts of allergens on the skin. Usually it's going to be on your back or maybe your arm, and they're going to prick or scratch the surface. And if that particular allergen is going to develop an allergic reaction, so they're looking for redness, swelling, or itchiness at that test site, then they can identify that as being an allergen that you're allergic to. And some of the things they might put on is like pollen, pet dander, dust, or certain foods. They might offer you a food challenge, and this just means you're going to take small amounts of food that you think you're allergic to and see what happens. It's commonly used to diagnose food allergies. It's not the most scientific way, but it is a way to start with testing for hypersensitivity reactions or type 1 hypersensitivity reactions. We can also get lab tests, and the one we're looking for is a blood test that's going to look at the IgE, and we're looking for IgE antibodies in the blood, and this can tell us if there's an allergic response or not. It can also help to rule out if it's an allergy versus something else. With type 2 hypersensitivity reactions, natural killer cells are being released, and they're going to destroy target cells. But we have also the autoantibody that binds to and activates receptors. And thinking an example of target cell malfunction in Graves' disease, it's going to target the thyroid. There are 5 main mechanisms through which immune responses can cause damage. It could be cell destruction, particularly IgG and IgM. It can be from cell destruction through phagocytosis. So macrophages and neutrophils can recognize and engulf cells that are coated with the antibody. Tissue damage from the immune complex disposition. And so the immune complexes can deposit into the tissue. It's going to trigger inflammation. It's going to lead to the damaging of that tissue. Again, we talked about lupus and rheumatoid arthritis. And then we've already talked about the antibody-dependent cell-mediated cytotoxicity, or ADCC, when we talked about natural killer cells, and then target cells like the TSH on the thyroid. In type 3 hypersensitivity reactions or immune complex mediated reactions, we have an antigen-antibody complex which is formed, and subsequently they're going to deposit into various tissues and in vessel walls and even in the extravascular tissue. And this immune complex will deposit the antigen that's going to trigger the inflammatory response, and it leads to tissue damage. And usually it's going to be IgG or IgM, and the complexes can circulate within the bloodstream and then deposit into the walls of the blood vessel and the extravascular tissues throughout the body. The deposition of the immune complexes will then trigger the activation of the complement system. And then therefore it's going to recruit immune cells and neutrophils and macrophages to the site, and it will include lysosomal enzymes which can cause tissue damage. It's also the immune complex mediated hypersensitivity reaction is not going to be limited to specific organs or tissues. It can affect multiple organs, multiple systems, and lead to a wide range of clinical manifestations. Serum sickness occurs after the administration of certain drugs or foreign proteins, and it results in a formation of immune complexes and subsequent symptoms. So you might see fever, rash, joint pain. And this is an example of an immune complex mediated reaction. Also, the Arthus reaction is another example, and this occurs when localized immune complexes trigger this inflammation and tissue damage. It's usually observed in response to repetitive exposure to a specific antigen. And then lastly, the type 4 hypersensitivity. It's a cell-mediated hypersensitivity reaction targeting the cellular-mediated adaptive immune system. and the cytotoxic T lymphocytes. And so with the cell-mediated hypersensitivity reactions, it's going to primarily involve the cytotoxic T lymphocyte, but it can also target T1 and T17 cells. So that's very specific. You don't probably need to understand it to that level, but it's the cytotoxic T cells, and they're recognizing specific antigens and initiating an immune response. In cell-mediated hypersensitivity reactions, so these T cells are directly killing the target cells that display the specific antigens. So on the other hand, they produce the lymphokines that are going to recruit and activate the phagocytic cells, such as the macrophages and neutrophils, to the site of the inflammation. Graft rejection is an example. This is when the transplanted tissue is being recognized as foreign, and so your immune system is going to destroy it. In terms of a TB test being an example, you might see a more delayed hypersensitivity reaction, and this is response to the intradermal injection of PPD, and that's why they call it a PPD test, the purified protein derivative, and this is derived from tuberculosis bacterium. And so that allergic reaction from poison ivy or metals is also an example of a cell-mediated hypersensitivity reaction with T cells that react specific to that antigen, and it's going to trigger that inflammatory response. So in terms of what's being tested on, I just want you to know the different types of hypersensitivity reactions. Give me an example and what's being targeted causing that reaction. Just in general, the most common hypersensitivity is an allergy, and it's usually a type 1. Environmental antigens can cause atypical immunologic responses to a genetically predisposed individual for pollens, molds, fungi, foods, animals, animal dander. And in individuals with this genetic predisposition, this allergen is going to trigger an abnormal response, and it's usually the production of IgE antibodies against the allergen. When we get re-exposed to the allergen, that antibody, the IgE, will bind to receptors on the mast cell and the basophils, and remember, those are immune cells, and they're going to trigger histamine, leukotrienes, cytokines, and mast cells and basophils. So what are you going to see? Itchy, itching, swelling, redness, sneezing, wheezing. Sometimes it can cause nasal congestion, even hives or urticaria, and in severe cases, anaphylaxis. In some cases, an allergen may be contained within a particle that's too large to be phagocytosed by the immune cell, and so they're protected by a non-allergenic coat. And this can prevent efficient clearance of the allergen and then prolong the immune response and make it last longer. Anaphylaxis, this severe, potentially life-threatening, immediate hypersensitivity reaction, is rapid and severe. It can be systemic or cutaneous, meaning it can be the whole body or just in various ranges of symptoms in specific areas. Usually the manifestations are going to be difficulty breathing, wheezing, swelling of the throat and tongue, low blood pressure, dizziness, rapid heartbeat, GI disturbances, and loss of consciousness. Or you might see cutaneous symptoms like hives, itching, flushing. Those are probably going to be more prevalent. Anaphylaxis is triggered by various allergens. It can be commonly an insect sting, certain foods like peanuts, shellfish, or eggs, or penicillin or aspirin in terms of medications. And some people can have anaphylaxis to latex. Desensitization is a process in which individuals with a known allergen are gradually exposed into small amounts of allergens under medical supervision. And this process will aim to reduce the severity of allergic reaction over time. However, the risk for desensitization is that it can trigger anaphylaxis, and that's why you need to be under the care of a trained healthcare professional in a very controlled setting. Autoimmunity refers to a condition in which the immune system mistakenly targets and attacks the body's own cell and tissues as a foreign invader. Again, we talked about this in terms of being influenced by a combination of genetic, environmental, and random factors. Certain genes are predisposed to autoimmunity, but environmental triggers such as infections, toxins, and hormonal changes can also play a role. The breakdown of tolerance, this is talking about, tolerance being the ability of the immune system to recognize and tolerate self-antigens. And this is going to prevent the immune response against your cells. But in autoimmunity, there's a breakdown of this tolerance mechanism, and it leads to immune systems being mistakenly targeted as self-antigens, as if they were foreign antigens. In autoimmunity, the immune system recognizes self-antigen as foreign and mounts on immune responses against them. They can result in chronic inflammation, damage to various organs and tissues in the body. They're not normally seen by the immune system, so in some cases, the self-antigens are targeted in autoimmunity and are not normally seen by the immune system during development. The self-antigens become exposed to the immune system through tissue injury, infection, or other factors, and they will then trigger an autoimmune response. So certain infections can actually trigger an autoimmune response. For example, rheumatic fever. It's caused by a bacterial infection with Streptococcus pyogenes, and then it leads to the development of rheumatic heart disease, which is an autoimmune condition. Also, infections such as viruses or bacteria can trigger autoimmune reactions that result in conditions like glomerulonephritis that affects the kidneys. They affect different organs and tissues in the body, and it can have a wide range of clinical symptoms and presentations. A more notable example of autoimmune disease includes lupus, rheumatoid arthritis, multiple sclerosis. Type 1 diabetes is also considered an autoimmune disorder. Hashimoto's thyroiditis are also examples of autoimmune diseases. So again, make sure you can provide some examples of each type of hypersensitivity or allergic reaction. These next slides give you a little bit more about the specific disease processes in the autoimmune examples. However, I'm not going to test you on the specific disease processes. You'll be asked to know them in depth as we go through the systems in which they are part of. But systemic lupus erythematosus, or SLE, is multisystem. It's an inflammatory disease, and the autoantibodies that are being mistakenly targeted are the nucleic acids, the erythrocytes, coagulation proteins, phospholipids, lymphocytes, and platelets. And so what you might see is joint pain, swelling, skin rashes, that characteristic butterfly rash across the face, fatigue, fever, muscle pain, kidney problems, lung involvement. You might even see neurological symptoms and hematological abnormalities as well. And people that suffer from SLE, they'll have periods of active disease or flare-ups, and then periods of remission when they don't have any symptoms at all. We will diagnose and treat SLE based on the clinical manifestations, an evaluation of lab tests. So we're going to do autoantibody testing and imaging studies. And we try to manage the inflammation mostly, and we're trying to prevent organ tissue damage. It is more commonly found in females. I think a very noticeable person or noteworthy person with SLE is Selena Gomez, and she actually required a kidney transplant because of the way that the lupus affected her kidneys and caused lupus nephritis. And here you can see the different types of clinical manifestations that I just talked about, but the percentage of what you might see in the different types of individuals. So actually, for Selena Gomez with renal disease, it really only affects about 50% of people with SLE. So most of the time, it's the arthralgias, arthritis, and the rashes that you'll see. And here are some another slide of just the common findings and what we've talked about already. So the lab test, the autoimmune tests that we're looking for, the antibodies, are the ANA, which is anti-nuclear antibodies, and that is part of the diagnostic criteria for lupus. In terms of alloimmunity, we talked about transfusion reactions, and I gave you just a glimpse of that in talking about the blood group system, the ABO, and the presence and the absence of a specific A and B carbohydrate antigen that is found on the erythrocyte or the surface of the red blood cell. And these antigens are genetically determined. So you're either going to be A, your B, AB, or your O based on the combination of these antigens that are expressed on your erythrocyte. When there's an incompatibility of blood transfusions that result in an antigen-antibody reaction, what is occurring is, so for example, type A individuals, they have an anti-B antibody, and type B individuals have an anti-A antibody. Type O individuals have both anti-A and anti-B antibodies. So individuals have naturally occurring antibodies in the plasma against the antigens that they lack on their own erythrocyte. When incompatible blood types are mixed, the antibodies in the recipient's plasma react with the antigen on the donor's erythrocytes, leading to agglutination or red blood cells. This agglutination will activate the immune system and result in the destruction of transfused erythrocytes. When we have a blood transfusion that needs to be performed, we have to make sure it's compatible with the ABO blood types. And the general rule is to transfuse blood from donors who have the same or compatible antigen as the recipient. So for example, individuals with blood type A can receive from blood A or type O. Individuals from B can receive from B or type O. Type O negative is considered the universal donor because it doesn't have an A or a B antigen on the erythrocyte. So it can be transfused to anybody, A, B, AB, or O, and it's not going to cause an ABO incompatibility. On the other hand, if you are type AB positive blood, you are considered the universal recipient because individuals with this blood type can receive transfusions of any ABO blood type without experiencing an incompatibility reaction. The Rh blood group system is another important blood group system that most of us forget, but it's primarily concerned with the presence or the absence of the Rh antigen. This is also known as the D antigen, which is a little confusing. But the Rh antigen on the surface of the red blood cell or the erythrocyte can also cause a response of incompatibility. So that Rh antigen, this is a protein expressed on the surface of your erythrocyte or your RBC, and the most significant Rh antigen is the D antigen. But there are other Rh antigens such as the C, the E, the little e, and the little c. The presence of the absence of this D antigen determines whether you're going to be Rh negative or Rh positive. So Rh positive and Rh negative, let's just talk about that now. If you have the D antigen on your erythrocyte, you're considered Rh positive. So you can be type A plus, B plus, AB plus, or O plus. While individuals lacking the D antigen are considered Rh negative. So they're going to be A neg, B neg, AB negative, O negative. So it just depends on your Rh antigen or Rhd antigen. So Rh compatibility is crucial during blood transfusions. If you're Rh negative, you can receive blood from Rh negative or Rh positive donors. However, if you're Rh positive, you should only receive blood from Rh positive donors. Otherwise, you're going to see hemolytic reactions occur. And the hemolytic disease of the newborn, this is also known as Rh disease. This occurs when mom is Rh negative, but baby is Rh positive. And so during pregnancy or delivery, the mother is exposed to the Rh antigen from the fetus's blood, and then her immune system produces anti-Rh antibodies. So in subsequent pregnancies, when the Rh positive fetus, if they have another Rh positive fetus, the antibodies will cross the placenta and will attack the fetal red blood cells. And this can lead to hemolysis and potential complications for the newborn. So this person has to get immune globulins that are Rh negative to prevent the production of anti-Rh antibodies and subsequent hemolytic disease of the newborn for their subsequent babies. And then lastly, the transplant rejection. We have hyperacute reactions that occur immediately or really shortly after a transplant, but it's very rare, and usually it's caused by pre-existing antibody in the recipient's bloodstream, and it's reacting to the antigen of the graft organ. An acute reaction, this is going to occur within weeks to months of the transplantation. It is cell-mediated. It's immune response that's primarily targeted. It's targeting unmatched human leukocyte antigens, or the HLA antigen, and this is the protein responsible for immune recognition. The recipient's immune system recognizes the transplant organ as being foreign, and it's going to mount the immune response to destroy it. The acute rejection can be managed with immunosuppressive medications to suppress the immune response. And chronic rejection, this is months to years, and it's believed to be a result from a weaker, ongoing cell-mediated immune response against minor HLA. Again, that stands for human leukocyte antigen. And other factors such as chronic inflammation and fibrosis. So chronic rejection leads to gradual and progressive damage to the transplanted organ, and eventually it's going to result in organ failure. Managing chronic rejection is very challenging, and the long-term outcomes of treating a patient with chronic rejection is quite poor. But overall, these classifications are going to help us to understand the different immune mechanisms that are involved in transplant rejection, which is a form of alloimmunity.